Hello everyone, this video from Destiny of Biological Sciences. Today we'll learn about the biotechnology principles and processes for NEET exam. If you are new in our channel, please subscribe our channel and you will get benefit from this channel. Now today we'll learn about biotechnology. So what's biotechnology is? So biotechnology actually deals with the technique of using living organisms or enzyme from organism to produce product and processes useful to human. মানে বায়োটেকনোলজিকে আমরা কাজে লাগিয়ে কি করতে পারি না মানুষের বেনিফিটে সহজে কম সময়ে কম খরচে কিভাবে ভালো প্রোডাক্ট তৈরি করা যেতে পারে সেই প্রসেসটাই হলো বায়োটেকনোলজি। সো যেখানে কি হচ্ছে বায়োলজি প্লাস টেকনোলজি, বায়োটেকনোলজি অ্যাকচুয়ালি মিনস biology plus technological application, technology. So here European Federation of Biotechnology given a definition, and the integration of natural science and organism cells parts thereof and molecular analog useful for product and services, analogs for product and services. Right now we can come here is the biotechnology. So before the modern biotechnology came, there is a traditional biotechnology where the organism involves is microbes involves. Production is low scale productions. Nowadays the production is going to increase by use of modern biotechnology where we use genetically modified organisms, in vitro fertilization technique, in synthesizing genes, using it a copy of it, developing a DNA vaccine and correcting a defective genes. These are the application of different areas of modern biotechnology. Now we learn about the differences between the genetic engineering and bioprocessing engineering. This content actually made according to NCERT class 12 syllabus, because most of the question is coming from NCERT. So that's why this combination exactly from NCERT. Whatever the sequence NCERT maintained, here also we are trying to maintaining the same sequence. Now will that the principles of biotechnology and core biotechnology, what's the difference between genetic engineering and bioprocessing engineering? Now we can see that the definition of genetic engineering, here the techniques to alter the chemistry of genetic materials to introduce these genes in host organism and thus change the phenotype of the host organism, like in creation of rDNA or recombinant DNA, gene cloning, gene transfer. These are part of, here we see that these are part of genetic engineering. Here we can see that bioprocessing engineering, maintenance of sterile ambience in chemical engineering process to enable the growth of only the desired microbes, eukaryotic cells in large quantities. Here we only maintaining during the process sterile condition where no other bacteria, no other microbes can grow there. Bioprocessing engineering, here like in manufactures of biotechnological products include the antibiotic production, vaccines production and enzyme production. These are different parts of bioprocessing engineering. Now we come into the what's the advantage of biotechnology over the other techniques that's available. Right now here we can see that different methods here available are. First is that we can see the asexual reproduction. Here asexual reproduction, sexual reproduction, traditional hybridization technique and genetic engineering. So all of them have some advantage and some disadvantage, and ultimate which have overcome this the genetic engineering. So what's in asexual reproduction? Here asexual reproduction actually is also known as vegetative propagation. Here the advantage is that preserve the genetic materials. So here is no variation occurs. So this also is one of the important disadvantage for asexual reproduction, because if there is no variation, the survivability with the environment, environmental changes can disrupt this process. If the environment change abruptly, then asexual reproduction get a problem. So that's another process is that sexual reproduction provides opportunity for variation and formulation of unique combination of genetic setup, some of which may be harmful to the organism as well as the population. Some of will be harmful to the organism as well as the population. Traditional hybridization technique used in plants and animal breeding. Traditional hybridization technique used in plants and animal breeding very often leads to inclusion and multiplication undesirable genes. In case of if with this here the sexual reproduction, what is the advantage of sexual reproduction? Sexual reproduction provides provide variation because there is a mix up of genetic materials and that leads to variation. In case of disadvantages কি হচ্ছে organism এবং population জন্য ক্ষতিকারক. Traditional hybridization technique used where it is used in plants and animal breeding. In traditional hybridization technique used in plants and animal breeding very often lead to inclusion and multiplication undesirable genes. In case of traditional hybridization, what's made possible? Inclusion of undesirable genes. Suppose I want two varieties, like in case of one is suppose one cow varieties this is there where one produce milk varieties good milk quality, another is the good disease resistant property, disease resistance. Now I want to hybridize these two character and get the hybrid one, but here the problem is that with this desired character some undesirable characters may also came up with this hybridization technique, and this is the big advantage or big disadvantage, not advantage, which is big disadvantage of hybridization technique, traditional hybridization technique. So that's why this hybridization technique this hybridization technique should have some superior or some other techniques should have there. The other technique is genetic engineering, which overcome this hybridization problem, allow us to isolate and introduce only one set of desirable gene without introducing undesirable gene into target organisms. Target organism এর মধ্যে কি হয় আনডিজারেবল জিনস ইন্ট্রোডিউস না করে ডিজারেবল জিনসকে আমরা কেবলমাত্র ট্রান্সফার করতে পারি জেনেটিক ইঞ্জিনিয়ারিং মাধ্যমে. দ্যাটস ইজ দ্য বিগ অ্যাডভান্টেজ অফ, দ্যাটস ওয়াই দিস জেনেটিক ইঞ্জিনিয়ারিং টেকনিক ডেভেলপ, রাইট? Now we come into the some terminology used in genetic engineering. Some terminology include some terminology we'll see here. First is cDNA or complementary DNA. cDNA or complementary DNA. Complementary DNA. Then we'll learn here different nuclease enzyme which degrade the nucleic acid, like the DNA manipulating enzymes. Nucleases two type, endonuclease and exonuclease. Endonuclease and exonuclease. Then we learn about our restriction endonuclease or RE enzyme, or these are restriction endonuclease. Then we'll learn here about rDNA or recombinant DNA. Learn about rDNA or recombinant DNA. Recombinant DNA or rDNA. We learn here the DNA ligase, different enzymes, DNA ligase, enzyme, properties of DNA ligase, how DNA ligase works, different types of DNA ligase enzymes. Now, then we hear about concept about plasmid, concept about vector. Then we learn about different methods like PCR method, DNA replica plating, techniques, then alkaline phosphatase, use of alkaline phosphatase enzyme, role of alkaline phosphatase, role of T4 polynucleotide kinase, enzyme, alkaline phosphatase, T4 polynucleotide kinase, alkaline phosphatase, T4 polynucleotide kinase. We learn these different techniques, different terminology here. Now we'll move the cDNA. What is cDNA or complementary DNA? Complementary DNA where RNA by reverse transcription it produces cDNA or complementary DNA. cDNA in terms it is in small letter cDNA or complementary DNA. It's a to make a DNA copy of mRNA. একটা mRNA থেকে DNA তৈরি হওয়ার পদ্ধতিকে আমরা বলছি cDNA. Actually, most of the biotechnological application this cDNA actually use because cDNA lacks intron. cDNA lack intron. cDNA তে কি থাকে না? ইন্ট্রন থাকে না। cDNA lack intron. cDNA কি দিয়ে তৈরি না only exon দিয়ে তৈরি। restriction enzymes, what is restriction enzyme? Restriction enzyme or RE enzyme, this is to cut DNA at a specific site. DNA কি করতে পারি একটা specific site এ কাটতে পারি making small fragments, ছোট ছোট ফ্র্যাগমেন্টকে ডিএনএ কাটার জন্য যে এনজাইম ইউজ হয় সেটাকে আমরা বলি রেস্ট্রিকশন এনজাইম। যে এই যে ডিএনএ কাটার জন্য যে এনজাইম সেটা এই জন্য আরেক নাম কি মলিকুলার সিজার অর সিজার অফ ডিএনএ অর নাইফ অফ দ্য ডিএনএ বলেও বলা হয়। ডিএনএ ছুরি বা ডিএনএ কাঁচি বলা হয় এই রেস্ট্রিকশন এনজাইমকে, রেস্ট্রিকশন এনজাইম, রাইট। নাও দিস এনজাইম, এই এনজাইমগুলোর নাম কেন রেস্ট্রিকশন হয়েছে? এই এনজাইমগুলোর নাম রেস্ট্রিকশন হয়েছে কারণ এই এনজাইমগুলো কি করে না এটা একটা ব্যাকটেরিয়ার ডিফেন্স মেকানিজম। এটা একটা ব্যাকটেরিয়ার ডিফেন্স মেকানিজম, যে মেকানিজমের মাধ্যমে ব্যাকটেরিয়া কি হয় ব্যাকটেরিওফাজের ইনফেকশনের হাত থেকে নিজেকে protect করে। So this enzyme got their name restriction because this enzyme naturally occurs in bacteria where they prevent or restrict viral infection by degrading the invading viral DNA. Here কিভাবে না bacteriophage, this is a bacteriophage, bacteriophage can infect here and create infection into the, this is the bacteriophage, is the phage virus and this is the bacterial cells, bacteria here phage virus, this is a bacteriophage infect the bacteria and this the term restriction enzyme coined by the Ledberg and Messelson. এই রেস্ট্রিকশন এনজাইম টার্মটা কে দিয়েছিল লেডবার্গ অ্যান্ড মেসেলসন। এই ব্যাকটেরিওফাজ যখন ব্যাকটেরিয়ার ভেতরে ইনফেকশন তৈরি করে তখন ব্যাকটেরিওফাজ তার নিজের ডিএনএটাকে ভেতরে এন্ট্রি করিয়ে দেয়। ব্যাকটেরিয়া নিজেকে প্রোটেক্ট করার জন্য, ব্যাকটেরিওফাজের ইনফেকশন থেকে প্রোটেক্ট করার জন্য কি করে, এই ডিএনএ গুলোকে কাটার জন্য এক ধরনের এনজাইম সিক্রেট করে, যার নাম হলো রেস্ট্রিকশন এনজাইম।